Good evening, and would like to welcome all of you to Anselm House's 5th annual Elving Anderson Lecture in Religion and Science. My name is John Rogers. I'm the Development Director at Anselm House. I want to thank you for joining us for this important conversation this evening. Brian Bateman, our Executive Director, sends his regrets. He's ill today and unable to attend tonight. Anselm House was founded on the campus of the University of Minnesota in 1982 as the McLaurin Institute. Many of you remember that name. Our mission is to connect faith and knowledge with all of life. Faith and knowledge are sometimes seen as rival accounts of the world, especially when it comes to the topic of science. But we at Anselm House humbly submit that faith and knowledge are actually deeply compatible when properly aligned, and that this alliance provides all sorts of resources for the pursuit of the true, the good, and the beautiful in our world, and particularly to our case here tonight, the University of Minnesota. Elving and Carol Anderson embraced this vision with their entire lives. Elving Anderson, who we honor with this annual lecture, was a much beloved professor of genetics here at the University of Minnesota from 1960 until his retirement in 1991. Elving embodied our mission of connecting faith and knowledge, and he held the belief that they were not only compatible but closely related to each other. He spent a good part of his career working out that relationship in his own field of genetics. Some of you will remember that Elving passed into eternal life on the eve of the inaugural lecture in 2014. And many of you will also remember that his wife Carol has been a faithful and very engaged attendee in the years since then. As many of you are no doubt aware, earlier this year on February 2nd, 2018, Carol made her way home to the Lord. We will miss them both and are so grateful for the opportunity to commemorate their lives and legacies this evening. At this moment, I'd like to recognize Elving and Carol's son, who's in the audience with us here tonight, Carl Anderson. On behalf of Anselm House, our entire community, please accept our condolences, and thank you so much for honoring us with your presence here tonight. And it's now my privilege to welcome Andrew Hansen, our Programs Director at Anselm House, to the podium to introduce our speaker this evening. Thank you.
Thank you, John. I'm actually not going to introduce our speaker tonight, but I am going to welcome you. It's good to have you here. Thank you so much for coming. And thank you, John, for those comments. Tonight's topic is a significant one, and as I said, I'm glad that you joined us for it. As we're going to explore tonight, new technologies of gene editing that have been developed in recent years open a number of staggering possibilities. As Elving Anderson, who we just heard about, saw decades ago, such genetic technology holds the potential to do significant good. From allowing us to develop more abundant food sources, to helping treat genetic diseases, genetic engineering has the potential to be a tool for alleviating suffering and promoting human flourishing. And yet, as Elving also saw years ago, these scientific advances bring with them possibilities for negative consequences as well, intended ones and unintended ones. In his 1943 work, The Abolition of Man, C.S. Lewis argued that human mastery over nature, including over our own bodies, through technology can easily become mastery by the few people who control such technology over a far greater number who lack that control. With an eye to the eugenics movement of the time, the disastrous consequences of which were then becoming obvious in Nazi Germany, Lewis noted the immense power that reproductive technologies gave living generations over future generations. Since Lewis's time, breakthroughs in genetics, most recently technologies to directly edit the human genome, have only amplified our powers in this respect. And this makes it all the more important that we think very carefully not only about what we can do with this technology, but what we should do with it. What responsibilities come with stewarding such immense power. Last year's consensus study on gene editing by the National Academies of Science and Medicine called for increased public discussion of the ethical and policy considerations associated with gene editing. The committee that produced the report made clear that this discussion should not just be among experts, but should not just be experts informing the broader public of their conclusions, but instead the committee called for a genuine conversation among the stakeholders in this technology, which is all of us, experts and non-experts alike. For that reason, conversations like tonight's are important, where all of us can come together in an attempt to understand how these technologies work and what implications they carry. It's clear that the questions here extend far beyond the natural sciences. For this reason, we've also invited Dr. Jennifer Alexander, a historian of technology here at the U, to comment after the lecture from a historical perspective that might inform the conversation on what responsible use of this particular new technology might be. Before we introduce tonight's lecturer, I just want to mention that we will have time for questions at the end of the lecture, as well as a reception afterwards, just down the street at the Anselm House Study Center. So I will give you more information about that at the end, but there will be plenty of time for questions and conversations after the lecture. It's my pleasure now to welcome Dr. Alicia Chen, who is an associate professor here at in the University of Minnesota's Department of Genetics, Cell Biology, and Development, to finally introduce tonight's lecturer. Thank you, Lisa.
So thank you, Andrew, for giving me the privilege and the honor to introduce our speaker tonight, Jeff Hardin. So Jeff is visiting from the University of Wisconsin-Madison, where he is professor and chair of the Integrative Biology, Department of Integrative Biology, where he's been for the last 27 years. So I know Jeff in more of a scientific capacity. So I'm familiar with this research, which focuses on the understanding how sheets of cells migrate and reorganize during embryogenesis in a process called morphogenesis. This process is necessary for the development of an organism with a distinct body plan. So basically all your internal organs are inside, your head's up here, and your rear end is on the opposite end. So he has published widely in this field since prior to his time in Madison. So to give you a little bit of background, Jeff received his undergraduate degree in Michigan State University, so a Bachelor of Science in Biology and a Bachelor of Arts in German Studies. He then received his PhD in biophysics at Berkeley, which is when his interest in morphogenesis started. He then continued this interest in his postdoctoral training at Duke University. So this research that he's done throughout these years has been done in model organisms such as sea urchin and C. elegans, which is a soil nematode, because it is really too difficult and, more importantly, unethical to do such studies in human subjects. But his research has significant implications on understanding many human diseases, including cancer. And this is because, to quote him, you can study how a Toyota works. You can study how a Toyota works in order to understand how a Mercedes works. So we can use model organisms to better understand how the human body is formed and how it works. So Jeff is also well known in the teaching circle. In addition to having published multiple articles in teaching journals, he has also co-authored a cell biology textbook. And lastly, so in addition to his training in science, Jeff is also has a degree, a master's degree in divinity from the International School of Theology. And it is this interest in both the sciences and in humanities and faith that reveals the kinds of questions that plague Jeff's mind. Nothing too heavy, just existence, knowledge, how to discern truth and ethics. So without further ado, help me welcome Jeff Hardin.
Well, Lisa, thank you for that kind introduction. You know, I could stay an extra day and not give my lecture tomorrow in my developmental biology class, and you could do the whole embryonic anatomy thing. That was awesome. Thank you. Thank you. Well, it's a great privilege to be here. I've heard so much about Anselm House. It is a delight to already have had many significant interactions with Anselm House students, fellows, supporters, staff, and faculty who are friends of Anselm House. I hope you realize what a tremendous treasure you have here on the University of Minnesota campus in Anselm House. So thank you for being here. And Lisa is right. We are wanting to talk about a big topic. And I want to contextualize what we're going to talk about by just using my own life as an example of how much technology has progressed in the whole area of manipulation of human embryos. So this is just in my quasi-adult life. So since I graduated from high school, here's what's happened. So in 1978, the first test tube baby, in vitro fertilization. Shortly thereafter, we had surrogate motherhood in the 1980s. A host of reproductive technologies followed very quickly thereafter with exotic acronyms that I won't take you through. And then in 1997, we had this star, Dolly the sheep. She was the first mammal cloned by something called somatic cell nuclear transfer. A really surprising result for many of us who are wizened developmental biologists. On my own campus, Jamie Thompson developed the culturing techniques that allowed the culturing of human embryonic stem cells, along with an ethical controversy about whether it was appropriate to destroy human embryos in order to culture these cells. And that was in 2001. And the technological hits have just kept coming. It's been remarkable. It seems that every other week I'm introducing something new to my students in my developmental biology class about that has just hit the headlines. So let me give you a couple of examples of that. Human cloning was used to produce a genetically matched human embryonic stem cells in 2013. That same technology which allows cloning was used for reproductive purposes in cynomolgus monkeys. And that was in 2018, so just in January of this year. It's not very successful, but technically possible. These technological advances raise huge questions about whether science itself is actually adequate to adjudicate on the ethical uses of these technologies. So here's James Watson, not exactly the kindest, gentlest guy on the planet, but obviously a Nobel laureate for his work on the structure of DNA. And here's what he said about this. The belief that surrogate mothers and clonal babies are inevitable because science always moves forward represents a form of laissez-faire nonsense dismally reminiscent of the creed that American business, if left to itself, will solve everybody's problems. Science itself simply can't address some of the deeper issues that the science raises. And so we need to marshal other inputs to think about these things, and that's what we want to talk about together this evening. And I'm going to be addressing some of these issues from an explicitly Christian perspective. I happen to be a Christian, as Licia mentioned, and so I'll be, especially later in this talk, raising some issues that I think Christians should think about as they think about the use or appropriateness of these technologies. Why should a Christian think about these things at all, you might ask? Well, first, the 21st century, as I've already mentioned, is booming with biology. In fact, a lot of people are calling it the century of biology. And it's very likely that many people here or their loved ones will need to decide about ethically challenging technologies, therapies. More importantly, perhaps, Jesus of Nazareth said it this way in his famous Sermon on the Mount in Matthew chapter 5. You are the salt of the earth. So Christians have a mandate to engage the culture, to think deeply about the issues that are inherent in the culture of their own times. And so that saltiness, in part, I think, involves thinking about biotechnology. And we're here, of course, to honor Elving Anderson. I want to express my condolences to your family on your loss. I was hoping to meet your mom, and I was sorry that I didn't get that opportunity. Now, Elving and I have exactly coterminal careers. He retired in 1991. That's when I came to the University of Wisconsin-Madison. So Elving is somewhat of a hero to us, a younger generation of biologists who are Christians. And he did a lot of seminal thinking about genetics decades before most of us realized how important it was to do so. And so this is I can recommend this book on behalf of God, very interesting book that he wrote with Bruce Reichenbach. Elving had a really amazing way of cutting to the chase, getting to the deepest issues in which Christian faith impinges upon biology. So it's an honor to be here in a seminar series that honors Elving's memory. Elving was actually an expert on human genetics. Well, I'm not. Lisa told you the kind of organisms that I work on. That's the Toyota part of that little metaphor. But I think it's important that non-experts like me, like most of you, engage in these discussions. The eminently quotable G.K. Chesterton said it this way: if the ordinary person may not discuss existence, why should he or she be asked to conduct it? We are living through these times in which these technologies are going to transform our lives, and so we all need to think about these things. All right, so let's consider genome editing, and I want to talk about both human and non-human uses of this technology with you tonight. So part of this is going to be a little bit of a science education session. Part of this will be my own personal initial musings on some of the ethical implications, thinking from a Christian perspective about some of the issues we'll discuss. So first, what's genome editing? You may have heard a lot about it, but maybe you don't know exactly how it works. Well, let me review something I think all of us were exposed to in high school biology, and that's something called the central dogma of molecular biology. Pretty simple idea. DNA provides the information to make a molecule called RNA, and then there's the molecular machine that moves along that pink molecule in the middle of the slide and makes a protein. So what this means is that we can study a protein by working with DNA, and that's what most of us who work in the lab do all the time. Editing the DNA then means that you will permanently make changes in an organism's ability to make these sorts of proteins that correspond to the DNA. And these will be passed on to future generations. So if you want to use a technical term, these are heritable changes in the DNA. What's raised a lot of excitement about the ability to edit DNA is represented by this molecule here. This molecule is called Cas9. Cas9 is something, part of a system in bacteria called the CRISPR-Cas system. CRISPR is an acronym. I will spell it out this one time, and then I'm just going to say CRISPR after this. You can think about your vegetable drawer in your refrigerator if you want. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. These are pieces of DNA. This protein, the big blue blob in this slide, is a CRISPR-associated gene, Cas9. Cas9 is a protein that is programmed by small pieces of that molecule I mentioned a moment ago, RNA. And what it does is it targets a specific region of DNA and cuts it, and we'll see allows specific changes to that piece of DNA. Bacteria use this system to fight off viral infections. They basically retain a memory of previous viral infections of previous generations of bacteria. And so this is what they actually use it for. But people quickly realize this could be a powerful technology. And so here are the people who realized that. So on the left here are Emmanuel Charpentier and Jennifer Doudna, and they are one team that they worked on a lot of the basic biology of the system. And Feng Zhang at the Broad Institute, which is a joint effort between Harvard and MIT, he realized pretty quickly you could use this to do editing of almost any piece of DNA. And in fact, Zhang and his colleague George Church demonstrated that you could edit DNA using this system in 2013. So let me show you a little movie that shows how the system works. Then I'll see how well you're paying attention because we'll have a little review slide. And then we'll talk about what we can do with this. All right? So I think this will be loud enough. All right, ready? Here we go.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut, but the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
All right, so here's the quiz part. A pairs with T, G pairs with C. Oh, you guys are awesome. That's great. Okay. All right, so what is genome editing? Very simply, it's a way to make targeted cuts in DNA and then allow the DNA to be repaired to either break a gene or replace a piece of that gene with a sequence that the researcher provides. So there's an enzyme, that's the big maroon blob up there. It targets a specific site in the genome and makes cuts. It's molecular scissors. And then the cell has a mechanism for repairing breaks in DNA. Your DNA is being repaired moment by moment as you're sitting here in this lecture hall. And there are 2 typical results. If there's no extra DNA provided, then as you saw, when the cell tries to repair the break, usually it repairs it in such a way that the gene ends up being broken. But if we give a piece of DNA that the cell can use as part of the repair process, it can swap in that DNA that we give the cell. And this allows the possibility of replacing the original DNA. Now, there's one problem with this technology that's not mentioned in that video. There are problems with what are called off-target effects. Sometimes that maroon blob up there doesn't target the right piece of DNA and cuts another place in the genome. That's an unintended consequence. And that's an important safety issue if you're going to apply this in a human context. But I have to say the technology is moving at lightning speed. Things are improving, diversifying this technology. It's just hard to keep up. So that's the technology, and now let's think about some applications of this. First, let's consider why would we want to use this in non-humans. You saw there was a happy little mouse in that video. That's an organism that we might do this on. My favorite model organism, as Licia mentioned, is the same one she works on, a little nematode worm called C. elegans. And this is an embryo in which we've done gene editing on a protein that is part of a group of proteins that allow cells to stick together. That's the pink color in this diagram. So this is a genetically modified worm embryo. Fabulously useful in the lab. Also useful in agriculture. We're on the ag campus here in St. Paul, and it's often true that cows injure themselves. Juvenile male cows here have horns. These are CRISPR-edited cows, less injurious. They don't have horns, so they've been edited. So this has allowed open season on editing of organisms, both plants and animals, in agriculture, and it's a hugely important agribusiness opportunity. There are other possible applications for non-humans. Let's consider a pest that plagues humans, mosquitoes. It's possible to introduce into a mosquito a piece of DNA that targets a region of the mosquito's genome and also carries all of that machinery for doing the CRISPR-Cas9. And so you can make a piece of DNA and put it into the mosquito that matches a piece of DNA, cuts that DNA, but there's more. It also produces the machinery to keep doing this over and over. This is like a global search and replace on your word processor. And so eventually what happens is that a mosquito that has this specialized piece of DNA can drive away the normal copy of the gene found in the population of wild mosquitoes by mating with these wild mosquitoes, because all of the offspring, because of the way we do this experiment, spread this piece of DNA, sort of like a DNA virus. It's called a gene drive. If you're in a lab, you have to fill out lots of protocols saying that your experiments in the lab are not going to cause one of these. So this is a safety issue, but the intent here is to do this intentionally to wipe out certain populations of mosquitoes that cause malaria in particular. And a gentleman you may recognize is doubling down on this technology. So this is Bill Gates, and Bill and Melinda Gates, their foundation, is banking on this gene drive technology as one of the ways to wipe out malarial mosquitoes that carry malarial parasites. You may notice, if you can read up close it here, but the technology for extinguishing species is dividing conservationists. So this is not without controversy, to be sure. And what are the potential controversial issues here? Well, there's the law of unintended consequences. I'll call that the Jeff Goldblum problem. Anybody remember Jeff Goldblum from Jurassic Park? Life finds a way, yeah. And he's signed on for the next Jurassic World franchise movie, apparently, too. There are other questions, too. Like, once you start one of these, do you get a do-over? Can you reverse it? Well, there are some strategies for doing so, but there are a lot of questions about that. And in the US, this is, who's going to regulate this? Well, boy, it's just not clear. It turns out that in the US, at least 3 agencies, the Food and Drug Administration, the US Department of Agriculture, and the Environmental Protection Agency, all think they should have aspects of regulatory control. You can guess what that's going to lead to, at least until that chaos is straightened out. Well, there are other uses of genome editing in animals, particularly in pigs. So it turns out that pigs are a model system for doing xenotransplantation. The idea is that you could use a pig organ to transplant into a human host. But you need to engineer the pig so that they don't cause immune rejection issues when you put the pig tissue into the human host. And so you may have remembered in the video, we can rapidly edit multiple genes simultaneously, and that was done in pigs. In fact, George Church's group, the fellow I mentioned earlier in the talk, was part of doing this. And here are some of the happy little pigs here. Pigs have also been proposed for a more exotic use using this technology. And that is to take the pigs and edit a gene that's known to be required for making a certain organ, let's say a pancreas. Now the pig can't make its own pancreas. You might ask, well, why would that be useful? Well, imagine that we introduce some human cells into the pig. Then perhaps the human cells would form a pancreas composed of human tissue in this pig. That's called a chimeric embryo. Unless you think that that's excessively exotic, these experiments have been attempted. So here's a pig embryo, and the red cells here are human cells inside of this pig embryo. Now, this was not allowed to develop further, but you can see that this technology is moving very, very rapidly. So there are a lot of applications in non-human organisms, some not so exotic, some extremely exotic. But the title of tonight's talk, so that I avoid any accusation of false advertising, is about human genome editing. So let's talk about humans, and let's move on and do that. Well, this is already being done, in fact, successfully. So you may know that one of the treatments for cancer is to engineer your own immune system cells. A group of cells that are part of a group of cells called T cells is one example. And they have been edited using CRISPR. The idea is to genetically engineer these T cells so that they specifically attack your cancer. And this kind of immunotherapy is a very hot topic right now. A lot of people are very excited about it. A broader use of this technology, though, involves a technology that was developed by Shinya Yamanaka. He got a Nobel Prize for this work in 2012. The technology came on the scene in 2006. It's called making induced pluripotent stem cells. So the basic idea here is we can take any cell from your body and we can force your cells to make 4 proteins, which are typically called Yamanaka factors these days. This causes these cells to switch what they become so that they now become stem cells. So these are called induced pluripotent stem cells, and that's these purpley cells here. And by varying the conditions in which you grow these cells, they can be coaxed to become lots of different cell types. So these IPS, or induced pluripotent cells, can be directed to make specialized cells. And importantly, we can engineer them using CRISPR-Cas9 technology. So we could take cells from you, we could make them induce pluripotent cells, but if you had a genetic defect, we could engineer those cells to repair the defect, cause those cells to differentiate to make the kinds of cells that you're lacking because you have the genetic defect, and introduce them into you. So this has been proposed for therapies, and in fact, this has been tried in several clinical trials already. So here's my favorite recently. This is from November 2017. This boy here has a debilitating genetically based skin disease. IPS cells were generated from this boy. They were edited. They were coaxed to produce skin cells, and they were grafted over most of his body. And he was near death. Now you can see he's kicking a soccer ball in this picture. So it's really remarkable. Amazing. Now, one thing I want to point out is that the same therapeutic technology could potentially be used, at least in some cases, for enhancement. So you could administer engineered cells to someone who wants to beef up some part of their anatomy or has some other thing that they'd like to enhance, blood cells, potentially muscle, things like that. Those are more exotic uses of this technology, but recognize it's not necessarily just for therapeutic purposes. So this is really exciting. There are lots of potential uses of this technology. But what's got most of the buzz about CRISPR-Cas9 as applied to humans is editing the germline. Now that might not be a word you're familiar with. What does that word mean, the germline? Well, it's just the idea that the editing can be transmitted to offspring via eggs or sperm. So at some level of the process, we edit either all of your cells so that the cells that ultimately will make the eggs and the sperm also got edits, or we edit egg and sperm cells themselves before we put them together to do some sort of in vitro fertilization. Either way, the resulting organism will carry the changes in the DNA, and these will be, using that word I mentioned earlier, heritable changes. So that's where the controversy comes from, in particular. But it's pretty clear that this technology, at least at some level, is going to work, and pretty soon. Why do we think that? Well, here are a couple of those cynomolgus monkeys. These were CRISPR-edited monkeys, and you can see the date. This was 4 years ago already. And it was actually dirt easy. You could just inject a mix into an egg, you also introduce a sperm, and then look for embryos that had undergone gene editing. Now, there are issues with the efficiency of this and whether it's 100% perfect, and we talked about the off-target issues. But this works in a monkey. If it works in a monkey, it will work in a human. And in fact, in 2015, Chinese scientists reported that they had tried this in human embryos. At that time, the director of the National Institutes of Health, Francis Collins, said this. He said, the concept of altering the human germline in embryos for clinical purposes has been viewed almost universally as a line that should not be crossed. And so the NIH at that time reiterated its ban on editing human embryo DNA. And in fact, some of the kingpins of the CRISPR-Cas9 technology met for a summit in 2015 in December. I think part of the leadership there was Jennifer Doudna, who I mentioned earlier. And they came up with a few recommendations. First, they did not recommend that CRISPR-Cas9 should be tried for establishing a pregnancy. Main reason there is safety. Not clear that the off-target effects have been dealt with, whether it's efficacious, a number of issues, primarily related to the safety of the resulting embryo. Would it develop into a normal fetus and a child? They also said at that time, to move forward with this would require broad societal consensus. I'm not exactly sure what the criteria for broad consensus would be, but this is something that they mentioned. And they recommended cautious development of medical applications that cannot be passed on to offspring. So the iPS treatments or other similar technologies, T-cell technologies, those are examples of those kinds of technologies. That was 2015. Now jump forward to 2017, and Andrew mentioned the study done by the National Academies of Medicine and Science, and they came up with a different set of recommendations. They called for prohibiting any alterations resembling enhancement. And one way of thinking about enhancement is that you're not going to fix defective flavors of genes called alleles with anything but a different flavor of that same gene that is present at a high percentage in the rest of the human population. So that was their way of kind of defining what would represent a non-enhancement, just fixing a broken gene. What else did they recommend? Well, clinical trials of germline alteration might be permitted, but only for compelling reasons. They didn't really define well what a compelling reason might be, but one example would be couples who desired biological offspring for whom embryo editing is really the last reasonable option. Now we can debate whether there is a last reasonable option, maybe in the question and answer period, but this is one of the criteria set out by the National Academy study. So this kind of gives the yellow light to CRISPR-Cas9 as applied to embryos, and that's what this figure caption says here, and that's in 2017. Well, two of my colleagues, Dietram Scheufele and this colleague, Alta Charo, who's in our law school at UW-Madison, were part of that panel. Here's what Alta said at the time. It's essential for public discussion to precede any decisions about whether or how to pursue clinical trials of such applications. And we need to have them now. So this event tonight is part of implementing Alta's recommendation. We need to be involved in these discussions. That's why we need to be educated about the science and the implications of the technology. And why is that especially urgent? Well, after this report came out in February of 2017, in August, the Mitpolitov lab at Oregon Health Sciences University, who has been a world leader in a lot of the experiments with human embryos, reported a successful use of CRISPR to fix an inborn error that has to do with a protein that, when defective, leads to cardiomyopathy, a thickening of the heart muscle wall in teenagers. And often this goes undiagnosed until there's a heart attack on the athletic field associated with the teenager. So the name of the protein is not important, but this was an example of using this technology to try to fix a broken flavor and allele of a gene. So the technology exists. It may not be perfect yet, but there will be technical advancements. And so there's been a lot of discussion about where will the first CRISPR-edited baby be born. Well, this map shows some of the major developed countries that have the capability of doing CRISPR technology. So a number of European countries, Canada have legislative bans, so it's illegal to pursue the kind of CRISPR editing that would result in a baby. You notice this country labeled number 1 here has no such legislation. However, the Food and Drug Administration in this country prohibits the use of these technologies. So if you ever wanted to make money off of this technology, they would not allow you to pursue it. They would not give approval. China here is likely going to be one of the leaders in this technology. They have very liberal rules about the use of these technologies. So a lot of people are thinking that China will be the place where the technologies are developed the most quickly. So here's what the Food and Drug Administration said. They will not review research in which a human embryo is intentionally created or modified to include a heritable, there's that word again, genetic modification. Some people suggested, well, down the road, if different countries are pushing this technology, others have bans on the technology, this will lead to something called medical tourism, which already exists for other therapies, where people will go to various countries because they have the availability of the technologies. Hard to say, just recognize that that's under discussion. What else is under discussion? Well, maybe it's informative to think about who's leading the discussion. Do you recognize these guys? This is a guy who's got a security issue with the implementation of his social media platform. It's Mark Zuckerberg and his wife Priscilla Chan. They have created an enormous research enterprise headed by another C. elegans scientist named Corey Bargmann in the Bay Area. The Chan-Zuckerberg Initiative is designed to bring biomedical technology to bear to completely annihilate human disease. That's what they want to do. And you can see this slide. Can we cure all diseases in our children's lifetime? Very ambitious. So the thought here is that if we throw enough money at this situation in a free market system, that technology is going to prevail and it's going to prevent disease. Perhaps. But other people suggested that genome editing, because it's expensive, may actually lead to further inequities in society. Those who have access to the technology at an unfair advantage over those who do not. And who will decide who has access to these kinds of technologies? Not clear. Currently, much of the discussion is by extremely wealthy people, often in Silicon Valley. And I would argue we need to broaden the discussion. Well, I think the Chan-Zuckerberg Initiative is very well intentioned, but why is it going to be challenging for most genes to do any sort of repair using this technology? It's because most genes don't act as solo artists. Genes have complex effects on organisms, and often multiple genes are required to allow a particular process to occur in an organism. So the first example, the first of those two cases is what geneticists call pleiotropy. Say I have one gene. Well, it turns out that that gene does a lot of different things. Now, I may really want to be fixing one particular thing or enhancing one particular trait, but that gene has so many other effects, I can't separate those effects of the single gene. So there are many traits that would be affected. That's a problem. Conversely, for many other traits, the action of many genes is required. So that's something called a multigenic trait. So in this case, lots of genes are required to get a particular trait. So that one you could presumably use CRISPR for, because CRISPR is good at editing multiple genes simultaneously, but still is going to be very difficult. And our understanding of the genetic basis of most traits in ourselves as humans is very, very rudimentary. There's almost no characteristic where we have a one gene controlling one outcome situation. There are a few, some devastating diseases, but certainly most of the time that's not the case. Here's an example of a multigenic trait, autism. So you may know someone who is autistic, and it's clear there's a genetic underpinning to autism. There's clear there are other things going on too. This is a classic example of a multigenic trait. I happen to think about this one a lot because this is my son Christopher. He's 29. He has profound autism. And so it's just not going to be possible to wave a magic genetic wand over Christopher and somehow treat his disease readily. So you probably need to get out of the mode of thinking about some sort of gene, a single gene for the ability to play piano like Mozart, or to run a 100-meter dash like an Olympian, or to do anything else that you think is an important trait to have. Genetics just doesn't work that way. All right, so let's talk about applying germline editing to humans. So what are the key issues? Well, one of the key issues is identified by Jennifer Doudna. Here's what she said in discussing an interaction she had with a child who had Down syndrome. The mother of this child said, "There's something about him that's so special." The scientist, Jennifer Doudna, pictured here, tears up telling the story. It makes you think hard about what it means to be human, doesn't it? Yes, it does. And that's one of the key questions here. Because if we are trying to determine what constitutes an enhancement, what constitutes therapy, we must have in our minds, what does it mean to be human? Because we're either restoring an aspect of what it means to be human, or moving beyond what has typically meant to be human. Now, some people have radically different views from the one that I'm going to present a little bit later in this talk. Let's take Peter Singer, for example. His view is that embryos, human embryos, really don't require particular ethical respect. In this respect, according to Singer, experimenting on a human embryo is not to be compared in significance with experimenting on a living, sentient mouse. So for Singer, humans are not particularly worthy of ethical respect. They're just animals like all other animals, and they're on a level playing field with regard to animal experimentation. But for many of us, experimenting or trying to alter humans, especially the germline, is a big deal. So let's talk about some touch points that relate to the nature of humans. Well, let me suggest three. First of all, I think we need to think about embryos as patients. Are they the subjects of therapy? That would be what you do to a patient. Or are they experimental test subjects upon which we can perfect certain kinds of technologies? That will be required to develop CRISPR technologies for editing the human germline. There will be failure and loss. How about enhancement versus therapy? Well, we'll have to talk about that, and I've already mentioned that that's an issue about defining what is kind of the baseline for being human. And then ultimately, all of this comes down to answering an important question. What is the essence of being a human? And I want to raise a question about whether genetics is sufficient to answer that question. I believe it's not. And that leads me to thinking about genome editing as a Christian. So I want to say that many of these issues are shared between Christians and people who have other worldviews. I had a wonderful roundtable experience earlier today with a number of people from University of Minnesota faculty. That was a great experience to have that discussion. But I'd like to argue for a few principles that at least I'm mulling over as a Christian. And let's go back to Elving here. What inner resources, Elving asks, will individuals have for coping with future discoveries? It's sometimes claimed, that's a typo, it's sometimes claimed that questions of the future will be so unique that old values will be inadequate. But I've not found any basic questions that will not profit from consideration of a biblical perspective. I think that's good advice from Elving. He wrote that, by the way, in 1978. Remarkable. Well, what are some biblical parameters for thinking about humans? A piece of Hebrew poetry that I share with my developmental biology students every spring semester is this, Psalm 139, verses 13 and 14. You formed my inward parts, you knit me together in my mother's womb. I'm fearfully and wonderfully made. God expresses his care for each one of us even before we're born, while we are embryos. And we can say more about humans. Humans bear God's image. They are his vice regents, carrying out his kingdom purposes in the world. We learn that in the very first chapter of the Bible. In the image of God, he made them, male and female, he made them. And they're supposed to act as stewards, caring for the creation, tending the garden as part of that kingdom mandate. And that might include caring for living creatures. And that might also include thinking about genetic engineering. These are humans that procreate in one flesh relationships. We see that in the second chapter of the Bible, Genesis 2. And the results of those one flesh relationships are begotten gifts. Children are a gift from the Lord. Blessed is the person whose quiver is full of them, Psalm 127 says. In addition, humans, because they are all image bearers, they bear God's image, are deserving of protection, especially the weak. So here's the question. That sounds great, but a fundamental question is an embryo an image bearer? I think it's a hard question to answer as definitively as we would like. First of all, the human authors don't know anything about pre-implantation embryos, which are called blastocysts. The Bible is a pre-scientific document. We need to be honest about that. And that means its language doesn't resolve this issue with the certainty we'd like. And as a result of that, sincere Christians frankly disagree about the ethical status of early human embryos. But I think most Christians would affirm the following. Such passages may not establish when human life begins, but they establish God's care and involvement from the very beginning. And that leads me to a position which I will call the wisdom of reluctance. Leon Kass, a Jewish bioethicist, wrote an interesting article many, many years ago when Dolly the sheep was cloned. And the title of that article was The Wisdom of Repugnance. It was kind of the visceral reaction you have about Dolly, and you think about applying that to humans, and you go, bleh. Well, I want to say that there's some wisdom in reluctance. If the answers to these questions are slightly underdetermined by the biblical data, then a sensible course of action is caution. Here's what Lutheran bioethicist Gilbert Mailänder said. If we're genuinely baffled about how best to describe the moral status of that human subject who's the unimplanted embryo, we should not go forward in a way that peculiarly combines metaphysical bewilderment with practical certitude by approving even limited use for experimental purposes. Gill is saying here that there's wisdom in being reluctant here based on what we know. There are further reasons for reluctance from a biblical perspective. Christians recognize the tendency of humans for sinful behavior. The Apostle Paul says, "For all have sinned and fall short of the glory of God." We are all susceptible to those patterns of behavior. Even before the catastrophic events recorded in Genesis chapter 3, though, humans required restraint on their knowledge. God gives some instructions to Adam and Eve. There's certain pieces of agricultural products here, since we're in the ag campus, that you should stay away from. So there are limitations on humans even before they do some catastrophic act of disobedience, because they're limited. But it gets worse in Genesis chapter 3. Human beings are fallen or sinful, and that, as I said, extends to all of us. And that means that a radical remedy was required, at least that's what Christians affirm, and we just got through celebrating that in Holy Week in the Western Church tradition. And that is that Christ, Jesus of Nazareth, God in human form, died for us. That means it's serious business. And that kind of sinful behavior means that we as human beings have tendencies to misuse technology. And let me mention one form of that misuse, which ethicists often call commodification, making a commodity out of human embryos. Here's Francis again. Here's what he said. The application of germline manipulation would change our view of the value of human life. If genomes are being altered to suit parents' preferences, do children become more like commodities than precious gifts? Excellent question. And I think many people of religious faith, of various religious traditions, because of these considerations, are more skeptical than others in our general population about the use of these kinds of technologies. This is a poll from 2016 from the Pew Trust, and it polled the percentage of US adults in each religious commitment group who say gene editing is meddling with nature and crosses a line we should not cross. We can unpack what those words mean. We can do that in the Q&A if you want. But those with high religious commitments were the highest respondents agreeing with that statement that the gene editing would not be a good idea. Those who have a moderate commitment, lower numbers. Those with low religious commitments, extremely low numbers. So this is interesting. And I think at least from the Christian perspective, this is predicated on these theological concerns about humans, their limitations, and what the Bible calls their sinfulness. And there are reasons to be concerned about this, to want to restrain some of these technologies, and that is the very scientists, the brilliant scientists who've developed the technology, their natural impulse is to take the technology wherever it leads. Let's take George Church, a brilliant molecular biologist, absolutely brilliant. Here's what he said during the National Academy's discussions. If these fixes for severe diseases are shown to be safe and effective, why would small or large enhancements accompanying the fixes be unacceptable? So George here is suggesting, well, while we're in there, why don't we just do a little fixer-upper? And the reason he thinks that that's okay, I think the underlying reason is he has confidence that technology is a good thing and that it always brings good results. Now, an issue here, though, if you stop to think about it, is that the very things that lead to enhancement could, in theory, lead to de-enhancement, at least in the wrong hands. So there are a lot of people who don't think about that possibility. Maybe I'm conditioned to think about this because I just read Aldous Huxley's novel written in 1932, A Brave New World again, where technology is used for de-enhancement. Yeah, very interesting novel in light of what we're talking about tonight. Well, all this leads me to suggest we, as Christians at least, I as a Christian, have to think about this as a balancing act. We should seek to treat the embryo as a patient, an end, a begotten gift versus a means at all stages of development. On the other hand, the Christian impulse is to love, and that means to use technology to prevent disease, beneficence. I just said we should be wary of excessive technological optimism. And the fact is, these considerations are in tension with one another. So this creates a tension for Christians, I think, in thinking about these technologies. The last thing I want to leave you with is a quote from a hero of the Christian faith who passed away earlier in 2018. And that's the Christian evangelist Billy Graham. You may not know this, Billy Graham gave a TED Talk in 1998, and it's great. And in this TED Talk, Billy Graham says, the problem is not technology. The problem is the person or persons using it. And as Christians, I think we need to take that seriously. And that leaves me with a profound sense that we need to be very humble in thinking that we are going to do the right thing with these technologies. And let's go back to Elving. All of this must be tempered with the humility that there are limits to the changes genetics can bring. So it's my prayer that as we discuss these issues together, and as we discuss these issues as a wider society, we'll think about what it really means to be human. What are the appropriate uses of technology? What should we expect from technology? And ultimately, as a Christian, I want to affirm that the fundamental problems that we face as humans are not genetic. They are not biological. Our most profound problems are spiritual. And it's my hope that Christians and others can inject those ingredients into this discussion. Well, thanks for listening. This is at least some of my musings about this. And what we want to do now is turn it over to additional thinking about these topics. So thanks very much.
Thank you so much, Jeff. Before we move into Q&A, I want to invite Dr. Jennifer Alexander to offer a comment on tonight's lecture. And Dr. Alexander is an associate professor in the Department of Mechanical Engineering here at the University of Minnesota. She's a historian of technology and teaches both history of technology and science courses, as well as an engineering ethics course here at the U. We have often been in a position similar to this before, where we've developed a new technology, we're on the cusp of using it, and given her expertise, we have invited Jennifer to reflect on the history of how we have used technology in the past to help us frame this conversation in light of a historical perspective. So I want to thank you, Jennifer, for being here with us tonight.
Well, it's a delight to have had the opportunity to listen to Jeff and have such a wonderful guide into some of these questions. And I just have 3 points really to make. I'll tell you quickly what they are, and then I'll launch into them. I want to give you some idea that it is possible indeed to reject new technologies. I'm not making the argument that we ought to, but I want to give you a sense that historically it has been done and it can indeed be done. I want to argue that there is indeed room for a contradiction between faith and technology, if not between faith and science, certainly between faith and technology. And then I want to say that we should pay close attention, very close attention, to any kind of technology that gives someone the power to identify something as pathological. That gives someone the power to identify something as pathological. So I'll launch into those after a couple of beginning comments. I'm actually a real big fan of Billy Graham, and have been for a long time, but he's just plain wrong. Technologies do have values built into them. They express the wishes of the people who make them, and most often historically they express attempts to control changing situations and bring them into line with the way the world's intellectually understood, to try to find a tool that will implement an intellectual vision. So it's not just a matter of how people use the technologies themselves carry values and statements about human capabilities, about the material upon which they operate, and about the potentials they view and do not view as possible. So I would just immediately reject what Billy Graham says in that point, despite being quite a fan of his over many, many years. A couple introductory comments. One thing that's really interesting to pay attention to when talking about technologies and new ones is the glamour of expertise. Now we live in a society in which expertise is a little sullied at the moment in many circles, but nonetheless there is a glamour of expertise. There's a sense of something that is beautiful and perplexing, and the one who is able to manipulate it has a certain mystique and mysteriousness that often carries with it a certain authority. So that's something to be aware of. We should also ask the question, must we do what we can do? And ask that in many different situations. And then, to use Jeff's own language, we should ask, what is reluctance? He urges us to reluctance, a wise reluctance, and I think he's exactly right. But I don't quite think, Jeff, that adds to balancing as you get in the end. I think reluctance is a little more reticent than balancing. So the idea of rejection being possible. And here I'll just draw your attention to a word and a group of people you may have heard of and not know much about, which are the Luddites, who opposed the introduction of particular machines into textile production in England in the early 1800s. And they succeeded in rejecting and driving out a particular type of machine for at least a generation. And when the machine actually was called a gig mill, it raised the nap on luxury fabrics like velvet, for example, instead of being done by shearmen by hand across a long length of fabric. Produced inferior work for the first generation, etc., at greater expense. And when the Luddite movement was finally quelled, it took the military to do it. So it is, and they succeeded for a generation in holding off technology, so it is possible to do that. We know that we have rejected, right after World War II, as Jeff and I think Andrew also mentioned this, rejected the idea of eugenics. And for example, what used to be the Journal of Eugenics changed its title to the Journal of Human Heredity. And you can look in the vaults of any patent office in the world and see many wonderful, brilliant technologies that have never been picked up. So simply because something is there and has a bit of momentum does not in any way mean it has to succeed. It is entirely possible for technologies to be rejected and to be lost. I mean, think of VCR and beta. Think of that old debate some of you will recognize. The second thing I wanted to say is there is a contradiction possible between technology and religion or technology and faith. Most of the literature in this area deals with science and religion, not with technology, and it deals with science as statements of propositional truth. But that's not what technology is. It's not statements of propositional truth. And maybe there's no contradiction between faith and science at the level of propositional truth, but the minute you start having people implement ideas about how they think the world should operate, then you have possible great potential for a contradiction. The best example of this, I think, is using nature as a model for human ethics. The rejected systems of social Darwinism, for example, many of you will recognize this term, that the survival of the fittest and the weakest should be allowed to be weeded out by society and not protected, which many people argued in the early 20th century, and also in the 19th century, weakened society. That's at a level of a propositional statement of what happens in evolutionary development. Right? So is there a contradiction with faith? I don't know if there needs to be a contradiction with faith in terms of how you think God might have made the world work. But there certainly is a contradiction in terms of putting that into any kind of action. And the contradiction lies not at the statement of what is true or not about science in the world, but it lies at the statement of what is true about human responsibilities. And here I allude to a number of things that Jeff listed in one of his last slides. Love, care for widows and orphans and the weak and the stranger, to which we are commanded in the Old Testament and older, if you prefer, in the ancient Hebrew scriptures and the newer Christian ones. The last of these real points about being careful about anything that carries the power to identify something as pathological. And here we look at Jeff's distinction between what is enhancement and what is therapeutic. The idea of what is pathological is something to be removed. Georges Conguin, the sociologist, described it as anything that is twisted, crooked, or awkward is pathological. And you hear there the social sense of something that gets in the way is what is pathological. Historically, what kinds of things have been considered pathological? Feeble-mindedness, how on earth would you define that? You know, someone who seems not quite to fit and gets in the way. Alcoholism. Right? Twisted, crooked, and awkward. And the question that Jeff rightly raises is a really serious one. What does it mean to be human, and what constitutes the human? And if things get edited out of the human, what is the state? What is their status? Right? Are horns pathological on a bovine? Twisted, crooked, and awkward, and they get in the way. Right? So these technologies, gene editing technologies, carry with them the power to define something as pathological, as unwanted, and not just to define it, but to take action. And that's why it's not the technology that bears no blame. This is why the technology itself carries with it a judgment on what is normative and normal and what is not. And that is never a non-moral question. It's never a non-moral question. And Jeff, I think very rightly, drew our attention to the questions of the wealth and power of people who can be influenced and work on genome editing. Expensive business. Expensive to research. Expensive to use. Right? And so, as with many technologies, Andrew pushes me to say something about it historically. The question to be asked, I think, most fundamentally is, where is love? Right? And there is love in care. Barrenness is suffering. There is love in care for disease. Right? But our primary, and I should say not but our primary burden, I think, if we want to speak as Christians, is to speak for love. And then the balancing act really is one of cost. Right? You do what you're good at. You do what you can to help people within your realm. Within your realm. But keep in mind that we are first and foremost called to love and called to care for the weak, the widows, the poor, the orphans, the stranger. And if there's a worry about eugenics, and eugenics was not only a Nazi program. You know, the Germans modeled themselves in many instances on American practices, the Germans of the 1920s and into the 30s and 40s. If there's a worry about eugenics, and we should be concerned about that, it's at the level of defining what is pathological and what is human, and what is worth protecting and what is not. And Jeff's example of a Down syndrome child is a wonderful example. You know, human, yes, of course. Worthy of love, worthy of care. And what we need to watch out for is a technology that defines pretty much anything as no longer human or no longer worthy of care. And that's not only at the level of the whole organism, that's also at the level of various kinds of suffering and kinds of pains. You know, is it inhuman to suffer? I don't know. Suffering is huge. Suffering is true. Suffering is not a redeeming thing. Right? But suffering may well be human, given, as Jeff says, the human tendency to sin, to do evil, not to care for, as we should, our planet and our fellows. So a balancing act, I would go more with wise reluctance and wise slowness, right? And not be driven by the glamour of wonderful potential, but the knowledge that, you know, what are we called to love within our circle? Who are we called to help within our means and our grasp? So I'd like to thank Jeff again very much for this very useful and very thoughtful development by someone who is, you of course say that this is not what you primarily do, but for us you are the expert. And it is delightful to have a forum like Anselm House provides, a forum like this annual lecture, to come together and discuss these things as they ought to be discussed, because these decisions cannot be made only in laboratories and only in treatment settings. They have to be made in broader conversations before our God about whom we love and how we love each other. So I'll stop there. Thanks.
Thank you so much, Jennifer. Those words are sobering, and in thinking about this lecture, I was looking a little bit at the history of eugenics. There have been some concerns that gene editing could lead to sort of new eugenics, new tools to seek to select for certain attributes of the human race. And one of the things that I found was, in searching online, was a postcard from Charles Dite, who's a physician here in the Twin Cities, provided funding for the Dite Institute for Human Genetics at the university in 1933. So this is I think it's in the U of M's archives here, a letter from Dite to Adolf Hitler praising him for his eugenics efforts and suggesting that this would become, I think he said, the most advanced national program for human betterment that the world has ever seen. And it was just a sobering reminder to me that things that we see for enormous potential for the good can often have very different outcomes. He could not see further down from 1933. We, with the benefit of hindsight, can see that looked very different from a program of human betterment. And so, Jennifer, I appreciate those remarks. And we are going to have time for questions, a little bit of time here, and then we'll have more time for discussion afterwards. Jeff, if you would come back on stage. We've got a microphone that I think Danica is going to be bringing around. So if you have a question, if you could just raise your hand, and Danica will bring that microphone to you. I might begin with a question while we're getting the first question here. Sorry, Jeff, you're behind me. There we go. We'll trade spots. This quick change that we had between saying, so Francis Collin in 2015 saying, we absolutely will not edit the germline, to 2017, these recommendations, we will very cautiously consider editing the germline. What accounts for that quick change? Is it just different people? Have we moved as a society in 2 years in a significant way? What's your take on why the shift in 2 years?
Can I answer that question after I make a comment on Jennifer's comments?
Please do.
Thank you. So I think I wasn't clear in communicating something. The balancing act is how we feel about Christian impulses, not about the implementation of the technology. I just want to be clear about that. I'm a Gil Mylander guy, so that I don't see a way that we can implement these technologies in a way, currently at least, that would implement any sort of Christian vision that I can be on board with. For a number of reasons that I articulated. So I think maybe I evidently wasn't clear. No, I don't think so. So that's why we need each other. So that will help me for next time. Okay, sorry. So Francis Collins, he's the director of the National Institutes of Health. The body that was making the recommendations in 2017 was the National Academies of Medicine and Sciences. Different bodies. Francis is a Christian. I think I haven't mentioned that. He founded an organization that I'm involved with called the Biologos Foundation. He is a Christian at the highest levels of international science. And I think he's motivated by Christian concerns, the comment about commodification that I made. So Alta, my colleague from UW-Madison, she is very much more progressive, if you look at her track record in recommending implementation of technology. So I think what you saw in the National Academies report was an edgier group making recommendations, and they didn't think they were making a big change in recommendations. They clearly were, though, because they said we should cautiously move these technologies forward. No one had suggested that, at least in terms of germline editing of embryos before. So that was a new recommendation. So to me, that was a very big deal, actually, that they made that change. So I think that accounts for the difference. One thing maybe Jennifer can comment, but sometimes technologies are refused, but in other cases what we see is an initial period of reluctance and then a rapid widespread adoption of a technology. Like some people are not convinced the technology is useful and then they see that maybe it would be, and they quickly capitulate. So I don't know if that's happening now. I hope it's not, and that's why you guys as citizens I think need to weigh in on some of these things. Because there will be tremendous momentum among the people who talk amongst themselves about these technologies. The guys developing the technologies, we talked about the Silicon Valley initiatives and that sort of thing. And that tends to leave out the ordinary person. I gave a talk like this in Madison a few weeks ago, and a friend of mine who got a PhD in Spanish said, what can a poor old person like me contribute to this conversation? Well, I think if enough poor old people like us weigh in on these ideas, it will have an effect on national policy, and I think that's where we are right now.
Do we have a first question out there? I see Luke's hand.
I want to ask about actual policy. Obviously, this is a very complex issue with many factors weighing in from different schools of thought. But is it possible to simplify this entire discussion and shrink it down to just saying no? Is it possible to simplify that and, you know, go to a senator who doesn't know as much and say, just say no?
I mean, I think that's a safe course, to just say no. I actually don't think Elving would have recommended that. He was a little edgier than I am on the use of these technologies. I think if you read his corpus of work, I am much more pessimistic about the use of this technology for germline editing. I think the somatic cell editing for human patients, that makes sense to me, and you can regulate that in certain ways that are reasonable to me. And some of the uses that are already in play to me are sensible and purely therapeutic without any moving the needle on who's human and who's not, or any sort of question like that. I mean, to me, that's a possible approach here. I guess the question is, if you had a way to every embryo who is edited is successfully edited, and you knew that every time, it was a guarantee that, you know, the technology was so reliable that you could repair particular genes that only lead to debilitating death. Let's look at Tay-Sachs disease as a good example of that. It's a mutation in a gene that encodes an enzyme required for making the insulation wrapping around your nerve cells. We know exactly what causes it. We know people who don't have Tay-Sachs, all of us in the room, have a particular flavor of that gene. And if you could repair the defective version to to me, that's a correct version of the gene, the functional version of the gene, and you knew you could do that every time. We can't do that every time, we don't know that. But if you let's say, hypothetically, if you could do that, then some people would say, well, maybe in the extremely limited case, we could consider it. There are other ways to think about that. I mean, I would say there are other ways to avoid that situation that we could talk about that don't involve directly editing any embryos, and so I would prefer to take steps that would prevent the situation of having an individual that has that Tay-Sachs mutation as one example. But so I think that's a safe approach, the germline editing of embryos, just say no approach. That's something politicians can understand. Just say no. We could say that in 5 words or less, right? That's good for politicians, I think, sometimes. So that it's a nice, snappy, simple message. They don't have to wade into the waters of some of the things we're discussing. So.
I think there's
Hi, wonderful talk. I really liked a lot of what you had to say. One of the things that worries me is that the kind of values and conceptions that you're talking about that we need to have around this, you know, go far beyond considerations of utility, considerations of basic rights, and so forth. They require a pretty profound level of articulacy on human beings and so forth. I mean, you know, that you mentioned Cass's Wisdom of Repugnance article, and he says in there, you know, that we're going to be talking about you know, issues like cloning and so forth like that. We have to invoke conceptions of what it means, what the meaning of human begetting and belonging, you know, and things like that, which not only are very difficult for people who share a lot of presuppositions, like people in this room, like Christians and so forth, to articulate and be able to really speak to. But when you branch this out to the broader culture, there's almost no kind of articulacy on that. So there were no common ground to be found, and they consider conceptions like that, these deeper metaphysical conceptions, spiritual conceptions, and so forth, to be sort of matters of personal preference, and you know, and so forth. And so, but what happens when you don't have that kind of articulacy on these issues is that you default to a kind of vague utilitarianism, a vague rights language or something like that, but that does still retain significant normative force in society. And so what I see happening oftentimes is that when this becomes a dominant mode of discourse, this kind of utilitarian language, and it does sort of become normative for everybody, then those who don't buy into it are kind of regarded as, well, let's just say, I mean, evil perhaps, or at least just unwilling to do the kinds of things for the good of society.
They're labeled Luddites.
Right, yeah. So you see this in places like Denmark, Iceland, where they're talking about outlawing circumcision. They're talking about having this goal of ridding the society of Down syndrome by a certain date. And in places where they've legalized euthanasia and so forth, they're talking about like forcing doctors to participate in it, getting rid of conscience clauses, and things like that, because under the presumption that if you're not for these things, then you're sort of against human benefit. Where you're sort of like imposing your own personal values on the rest of society, which is, and so forth. So what do we do to combat that if society grows more and more and more towards saying that if you're not for these kinds of things, then you're a bad person, or, or something like that. So I'll just...
That was a lot of important things. A lot of important things. So sociologist John Evans at University of California, San Diego wrote a really great book called Playing God about the history of bioethics in the United States. And bioethics used to be populated by people who are kind of armchair ethicists like me, who were thinking about, maybe they were biologists thinking about the wider context, and many of them had religious sensibilities. They were forced out of the professional guild of bioethics. So now bioethics is mostly about making sure we have the right regulations in place, that we do things with a proper procedure. We're not asking the larger meta questions, is it right or is it wrong? So I think bioethics in the United States has lost something crucial, speaking broadly about it as a discipline. And if you're a bioethicist here, I don't mean to denigrate what you're doing, but I do think that Evans is right in that sociological analysis of that discipline. And so that's a form of pragmatism. It's a little different from utilitarianism as an ethical view, but those are cousins in a way because they look at what's practical, maximizing the good, supposedly, in utilitarianism. And in pragmatism, we just do what we can practically, and we kind of don't worry about the rest. And that's an oversimplification. But so what we can do about it, well, I think that's a big challenge. I mean, I think if people in the academy are willing to stick their necks out a bit and argue from general principles that can be shared by Christians and non-Christians alike about why this is a bad idea, why it's unwise, then that's a really great way to argue. I mean, I was at a table at the round table today with a person who described himself as a secular humanist and atheist, but he said, trying to meddle with our genomes, which evolution has fiddled with over millions of years, probably not wise. So we both come to a position where this is unwise from different starting points, but there's a common thread there. So we need to seek those commonalities in our culture. I think that's the only way, because if you simply say, well, based on my biblical argumentation, you should not do this, I've not found that to be an effective way to argue in the public square. And that used to be an effective way to argue in the public square in the United States, but no longer. We're not where Iceland is, but I worry. You may remember I have a profoundly autistic son, and he would be the subject of a eugenics purge probably in other kinds of cultures. So I'm extremely sensitive to that, and I totally agree with you on that.
We will take, this is the last question.
Oh, we gotta let Carl ask a question.
Plus Carl's question.
Thank you.
Okay, so that's interesting because I was thinking more pragmatically, biologically. If you know of, because I'm not in academic circles anymore, but if you know biologists that are making an argument, I mean, it seems to me like living things, they spend a lot of effort preserving genetic diversity and creating genetic diversity. And do you hear the argument that like doing this might affect our resilience as a species because we're going to lose some of that genetic diversity? Is anyone making that argument that you hear of, or?
Yeah, I think some people make that argument. The arguments about doing the gene drives, part of the argument there is precisely that argument. We don't really know. We have a bad track record at managing ecosystems. When we introduce species to try to fix something, usually bad, very bad. So I think the same argument can be made about the ecosystem within our genome, right? Do we know enough to do things? I think the answer is no, probably not. We don't currently know enough about that. You'd have to edit a lot of genes to affect the overall genetic diversity of our species. I guess that would be possible at some point. I think in these, right now it's maybe not possible, but we do need to think about the implications many, many years hence from now, because I think we'll look back at this era when we were discussing this as a pivotal time. That would be that's my guess anyway.
Well, thank you for your fascinating talk. One thing I'm wondering is, could you imagine any situation in which gene editing should be used to totally eliminate a human defect? If there was widespread agreement that this was a defect that should be eliminated, and if so, how would this be essentially any different from eugenics? And of course, we know eugenics means good genes.
Yeah, I think there's been a lot of discussion about that. Are there mutations that are in the human population that they're just bad? Like they have no redeeming value at all. I think the answer to that question is difficult. I mean, I really do. I'm guessing that will happen, though, just like we eradicated smallpox, and then there's a big discussion about should we get rid of the last remaining vials of frozen smallpox virus because it's bad, right? But then, oh, wait, if we ever need it again, then it's gone, and what are we going to do? So I think it's not the answer to that question is less obvious than you would think. Like you would think maybe there are genes where we should remove all of the bad flavors of that gene in the entire human population. That would require a massive project, I want to point that out, which we couldn't possibly do today. But yes, I worry about eugenics does mean good genes, but the problem with that is you can use that same approach for whatever gene you want to change the distribution of in the human population. And that's the just say no. That's where you want to say just say no, because we can't handle that. We're not going to be able to distinguish the one from the other. And that's aside from all of the embryo loss. If you were doing germline editing, you'd have to have some other way to do it probably than we currently have. So I think that is not possible, and probably not where we can't do that right now. And in order to get to the place where we would want to do that, wow, there would have been a lot of things that would have happened that I would be very uncomfortable with. That would be my own answer to that, but I know that there may be other people in the room who might disagree with that. Perhaps we can talk about that over at the Anselm House during the continued discussion.
Would you help me thank Jeff for the lecture tonight? Before we go, I have just a few quick announcements. Tomorrow evening at Anselm House, we're going to be hosting a reading of Dorothy Sayers' The Man Born to be King, which was a radio drama that was originally performed on the BBC that is a dramatization of the passion narrative of Jesus from the New Testament Gospels. So if that's of interest to you, Katie Ware is going to be facilitating that, who wrote her dissertation on that drama, so you'll be with an expert studying that. If you're a student at the U, I'd also invite you to join us tomorrow for our weekly espresso hour over at Anselm House. I'm going to say in a minute where you can find Anselm House if you don't know where that is. And I would also invite you to consider our Fellows program. We're taking applications for next year, and this is a year-long exploration of how the Christian faith connects to all of life, including the work and the study that you're doing here at the University of Minnesota. So if you're interested, you can find more at the table in back. And if you're joining us for the first time, Anselm House is new to you, I would invite you to come to one of our open houses. These are a chance to get to know more about us, what we do. The next one of those is coming up April 24th. It's a tight 1 hour chance for you to find out more about Anselm House, meet some staff, meet some students, and find out how you can get more involved. And last but not least, I would love to invite you all to come over to that study center immediately after the lecture. We're going to have some desserts and beverages. In order to get there, you'll go right out the main doors. You'll take a right to Cleveland Avenue, so the main street, and then you'll take a left, and you'll just go about 2 blocks down to Carter Avenue. And once you cross Carter, the house will be very obvious. It's got lights outside. It'll be lit up. There'll probably be lots of people by the time you get there. So it's a great context in which to continue this discussion. I know there were a lot of questions that didn't get asked, and we'll hope to see you all over there. Thank you so much for coming tonight.